Good morning students. Welcome to LEELA'S tutorial. In last class we have observed how we are going to culture the embryonic stem cells. Today we are going to see the culturing of adult stem cells. Right? Stem cells are undifferentiated mass of cells which have self-renewal ability, and also they have ability to get transformed or develop into pluripotent or the multipotent cells. Right? These stem cells are classified into two types, that is embryonic stem cells and the adult stem cells based on their origin. Right? So today let us see how we are going to culture these adult stem cells. First, the name itself implies that these stem cells are obtained from adult organism or any developed organism. Right? So here these adult stem cells are also referred as somatic stem cells or resident stem cells. So these somatic or the resident stem cells are present throughout the tissues of the human body or the organism. So if you take a human, so where you can find these adult stem cells, you can find the adult stem cells in skin, bone marrow, heart, liver, kidney, tooth bud, brain and spinal cord, and even in the rib cells. Right? So here the different parts, so in different tissues and different parts of the human, you can isolate these adult stem cells. Right? How you define them? Adult stem cells are undifferentiated cells which have ability to get transformed into any other type of cells which are present throughout the tissues of complete organism. Right? So in the complete organism, whatever tissue you take, in these tissues you can obtain these adult stem cells. So once you select the organ or the tissue from where you want to extract these or from where you want to obtain these adult stem cells, just select that organ. So if you are selecting the liver or the heart or the kidney, any organ, right? So first it is the biopsy and the tissue harvest. So what is this biopsy and tissue harvest? The tissue or the organ, whatever you are selecting, that should be cut into the minimum size tissue which is used as an explant, and that explant, by the surgical method you are going to obtain the tissue part, right, and then you are going to harvest that one or grow that one. So in order to grow any animal cell, the first process is disaggregation of the tissues. Right? So again, by means of your enzymatic disaggregation process, that is trypsinization, you are going to disaggregate the tissue. That means from the tissue you are going to separate the cells. So once the separation of the cells or the disaggregated cells are obtained, you are going to harvest them. Right? The harvest, and you are going to culture them in suitable animal tissue culture media. In order to obtain stem cells, you want only the stem cells now. You doesn't want to produce any specialized cells here. So first it is a biopsy, then tissue harvest, then after culturing you are going to isolate the stem cells. Right? Separate any other contaminating tissue or the contaminating cells and isolate only the stem cells from that culture. So once you obtain the stem cells, either you can grow them as anchorage dependent cells, that is ADCs, or anchorage independent cells. So here if you are going to grow them as anchorage dependent cells, when they have this property, when they have anchorage dependent proteins. So when due to the presence of anchorage dependent proteins, the cells will go and adhere to the substrate, that is the lining of the container. So because of which, what happens? They grow as a monolayer of a cell. They occupy the complete surface of the container or the vessel in which they are grown. Right? So here animal tissue culture flask consists of filter cap, right, and the suitable animal tissue culture media with all the nutrients required for the growth of your stem cells, and also they require 7.4 pH and the temperature 37 degrees, and also they require 4 5% of CO2, which can be exchanged through this filtered cap. So where you are going to incubate this? In the CO2 incubator, right? So after certain incubation, under favorable conditions, you obtain a monolayer of your stem cells. Otherwise, if the cells are grown as AICs, that is anchorage independent cells, that means here the anchorage independent cells means here they lack anchorage proteins, because of which they don't go and bind to the substrate. They are freely suspended in the animal tissue culture media. You can see here anchorage dependent cells, they are adhered to the substrate, right? They get adhered to the substrate, then only they grow as monolayer. Whereas here after inoculation, the inoculated cells are freely suspended in the media and then they divide, right? So here in case of anchorage independent cells, you are going to grow them as suspension cultures, where the cells are freely suspended in animal tissue culture media, right? So it is the cell expansion and differentiation. So what do you mean by cell expansion and differentiation? Till here what we call the stem cells are undifferentiated cells, which doesn't have any structure or function. But here they are gaining the function. So that means the differentiation process is occurred. That means the cells are proliferating or dividing, right? That is nothing but the cell expansion and differentiation. That means they are obtaining specific function in order to become a differentiated lineages. That is your precursor cells. They differentiate into precursor cells and then they develop into multipotent cells. The precursor are nothing but your pluripotent cells, right? The stem cells here, which they are having the totipotency. Totipotency is ability to the single cell having an ability to transform into a complete organism. That is your stem cell. Now the stem cell, after certain cell divisions, it has entered or it has become pluripotent due to differentiation. That means the pluripotent cells, they are called as lineages, right? That means these lineages have only ability to differentiate into specific type of cells. For example, if you say hematopoietic stem cells, in this case of hematopoietic stem cells, these are your precursor cells or pluripotent cells, which have ability to form all the cells which are present in blood. What are present in blood? Your RBCs, WBCs, platelets, leukocytes, everything. So all the cells which are present in the blood can be produced by this precursor cell. So we call them as lineages. So at this stage, the cells are undergoing cell expansion and differentiation to different lineages. So based on the cell you want to produce, you are going to select this differentiation media in which they form that particular. If it is another example, you can take the neural stem cells. If it is a neural stem cell, it have ability to produce all the cells, the nerves, everything which are present in the neural system. Is that clear? So like that, here you are going to allow the cells to undergo differentiation to different lineages. So as a result, you can produce whatever you want, the bones, cartilage, adipose or any other different types of cells based on your interest. Right? So that is here the stem cells after isolation, either you can culture them as monolayer of cells where they are anchorage dependent cells, or you can grow them as anchorage independent cells or the suspension cells. Right? So here when you are growing them as monolayer, the same monolayer will be used for further experiment or the treatment or whatever you want to use. Right? Whereas here in case of cell expansion, it is a time taking process where the cells are differentiating. Right? The cells are getting differentiated and getting developed into your precursor cells, that is the pluripotent cells, or the multipotent cells. Right? So here you can obtain a free suspension of cells. So either it is a ADC or the AIC, the cultures which you have produced, right? The adult stem cell cultures, these can be used for your cell therapy treatment, where you are treating the diseases using these stem cells. Right? So here it is called as cell therapy treatment. You are going to treat them by using the cultured cells. Right? So where you can use these cells, what diseases can be cured by using your adult stem cell cultures. So that is the spinal cord injuries. In case of spinal cord injuries, skin burns, osteoarthritis, fractures, muscle dystrophy, heart diseases, etc. So in all these diseases, you can make use of these adult stem cells, right? So here adult stem cells, they are also referred as somatic or the resident stem cells. Okay, so with this we are done with this stem cells topic, right? If you have any doubts in the topic, please put it in the comment so that I can answer. Okay, if you have any other queries, you can contact me. Okay, subscribe for further videos.